Pleasure to introduce the next speaker, Dr. Duccio Medini. Duccio is one of our scientific advisors, international scientific advisors. Duccio comes from the pharmaceutical industry. He was with Chiron, Novartis, and then GSK. He actually has a relationship with New Zealand stemming from his close involvement in the MEMS-B development back in the early 2000s. Those of you who remember, we had the outbreak of MEMS-B. And Duccio tells me how much it was a very positive experience working with New Zealand and the public health around developing the vaccine Bexsero, which is now the vaccine that's just used against meningococcus. I met Duccio as part of the R3 LEAP. R3 stands for RNA Readiness and Response. Duccio has led that major global initiative around the development of RNA technology, which he's going to talk to you about. The reason that I selected Duccio as a science advisor is there's nothing Duccio doesn't know about global RNA technology and the developments that are going on. The R3 LEAP was a consortium of 23 international groups right across the spectrum of RNA technology, from the manufacturing side all the way through to the development of novel products. And I'm sure he's going to give us a wonderful representation of what has been achieved through that and how we can use his knowledge of what is happening globally in the RNA space to the development of our New Zealand RNA platform. So I'd like to welcome Duccio to the podium.
Thanks a lot, John, Gary, Kirsten. Thanks a lot for having me here. It's really a fantastic opportunity. I think the platform that you guys have built is amazing. There's a lot of promise. And I wanted to use this time that I've been given today to share with you the story of the R3 program that has been my opportunity to get to know John and the team at University of Auckland. And this story started in March 2020, in a way. And I don't know how many of you are familiar with this picture here. Where am I? Where should I point? Oh, yep. Now it's better. This is a very sad picture for many Italians. This is a city in northern Italy called Bergamo. What you see are military trucks. Over the course of little over 1 month, Bergamo was the first city outside of China to be hit very badly by COVID-19 infection. And in one city only, over the course of the first month, there were more than 6,000 deaths. They didn't know where to put the cadavers. That's why they call them military. And the trucks that you see are cadavers being brought outside of the city because there was no place to put them. That's when R3, in a way, started. That's when Welcome Leap started thinking, never again. How can we use RNA to make sure that something like this will never again to us? And so, fast forward 1 year, now it's early 2021, we begin to see that RNA has the potential, it's actually demonstrated, not just the potential, to completely change the timeline of vaccine development from 10 years to 10 months or 11 months. And we also see the huge potential beyond vaccines. As we heard earlier today, the leading companies in the space were not created thinking of vaccines, actually, were created along therapeutic concepts. However, already now we have completely closed the pandemic phase, and still access to the COVID-19 vaccine remains extremely uneven. Low-income countries have not received the vaccines for more than 1/3 of their population. And the cost of the product and access to other products that can be developed on the technology remains very challenging. And the clinical development outside of the two leading companies that we have the fortune to have represented here, it's progressing very slowly. So this great promise of being able to do everything with RNA, it's actually progressing at a very slow pace. So we decided to ask a different question. What if we could have a distributed network of biofoundries implementing standardized technologies that could produce RNA therapeutics using the same processes for a number of diverse products, cancer, infectious diseases, vaccines of different kinds. And also, since it's a standardized technology multi-product, by the time the next pandemic comes, there's no adaptation that is required, there's no scale-up that is required. All these multicolor, multi-product biofoundries can all turn to red and can all manufacture the pandemic response product. In this way, you would have economically sustainable pandemic preparedness through something that is not dedicated to pandemics. It's dedicated to peacetime drug development, right? Now, why can't we do this today? What's holding us back? What's the reality today of biologics manufacturing, including RNA manufacturing? Reality is that we have multiple scales of manufacturing. Every batch-based process requires multiple scales to be executed. You go from in vitro, benchtop, early preclinical, late preclinical when you go in non-human primates, and then you do phase 1, phase 2, small-scale clinical, then you do large-scale with phase 3, and then you go to market. Every time you change scale, you have to tech transfer, scale up, re-optimize. And when you change product, again, you have to adjust the process. It might be a small adjustment, it might be a large adjustment that requires 6 to 8 months to tech transfer the manufacturing process for a new molecule, which means only if you have about $500 million, you can think about developing a product to commercial scale, which means you can only do that if you live in a vertically integrated company, where you have research, development, manufacturing, commercialization, distribution, marketing, etc. That's the only way that you can do biologics today. There's no other way. Now, the problem, if you think about it for a moment, is technical. It's not a financial problem. The problem is that product design depends on product manufacturing. So when you design a new product, you have to think, okay, how will I manufacture this new product? You don't have a standardized manufacturing that you can bring up to commercial, whatever product you design, that will take care of your design, right? You have a dependency between product design and manufacturing. So we asked, is this only a problem in biologics development? Are there any other industries that faced and solved this problem? And actually, there's one that we took inspiration from, and this is semiconductor industry. I don't know, how many of you have some familiarity with the history of semiconductor development? Okay, so up until semiconductors were invented in 1956, what you see in this chart is on log scale. No, this is not log anymore, sorry. You see the number of companies incorporated worldwide in any semiconductor-related activity, okay? Between 56 and 78, this number is 3 per year. It's not a very vibrant ecosystem, okay? Why is that? That is because, exactly as it happens for biologics and RNA today, in those days, when you designed a new chip, you had to think, okay, this is a new design, how the hell do I manufacture the new design? You had to calculate the quantum magnetic dispersion patterns of the electrons in the different layers of the wafer. You had to rethink it from scratch, okay? So if you were in Hewlett-Packard, IBM, you could do that. Otherwise, it was very difficult. Then in 78, something very important happened. And it's an event known as the VLSI revolution, Very Large Scale Integration Revolution. Two guys by the name of Carver Mead and Lynn Conway They came about with the Medini revolution, or the VLSI principles. They developed standardized rules by which if you would design a new chip according to those rules, you knew that there was a standard process that could manufacture it. You didn't have to adjust the manufacturing process anymore. You had to adjust the design to follow the rules, but once you did that, you were guaranteed you could manufacture. What happens? The number of product designers exploded, mostly in academia, because you didn't need to be in the industry anymore to be able to design. You just had to buy the book with the rules, follow the rules, and design. And shortly after, use a software that was developed in a DARPA program to do that instead of doing it on paper, right? And after 9 years, the pressure of new designs was so gigantic that the first, whoops, something strange happened. The first pure play foundry was created. What is a pure play foundry? A pure play foundry is what we would call today a CDMO. So it's a manufacturer that does not have any product in development. They don't have any design of their own. They just manufacture for others. This is TSMC in Taiwan. It's still today, they manufacture 45% of the chips of the world. And look at what happened with the red histogram thereafter. The number of companies that was started being incorporated exploded. Why is that? Because you have now two business models. One is the vertically integrated company where you have everything in one. That still is a good model. It still goes on. We still have Hewlett-Packard. We still have Intel. We still have IBM. But you also have these two separated models where you have pure play foundry, like TSMC, and then companies that only design, like Apple, like Broadcom, like NVIDIA. NVIDIA today is the company with the largest capitalization in the world. They don't manufacture a single chip. It's all manufactured elsewhere. Why is that? Because you have standardized manufacturing processes. You don't have to reinvent how you manufacture. So you can separate the IP, and that works very well. So this is the kind of revolution that we thought mRNA requires. Can I try? Yes. And so, to do this, we put together the team that we have been calling the R3 program for the past 3 years. About 150 scientists with 19 leaders scattered around the world, including John and Fiona and the great team in Oakland. 2/3 from academic organizations, 1/3 from commercial organizations, companies of different maturity levels, focusing on 3 areas. Develop the automated standardized RNA manufacturing processes that do not depend from products and that can work at multiple scale from preclinical to pandemic response without changing anything. Demonstrating these technologies on diverse RNA-based products, vaccines, therapeutics, viruses, bacteria, different things, different formulations, different deliveries, and developing the rules, developing what will give democratized access to this process is the set of rules that allow even a PhD student to design a new product knowing that it will be well manufacturable, right? So I want to go one by one into these 3 pillars, hoping to stay in the time that I've been given. First, manufacturing. Here's is where we saw a strong paradigm change, and this is what we usually call the shift from yogurt to flow chemistry. So what has yogurt to do with RNA? One of the key processes for RNA manufacturing is in vitro transcription, when you go from a DNA template to the naked RNA molecule. How is it done normally? This is done in a batch-based process, which means that you get a tank or a small plate, depending on the scale that you operate at. You mix your ingredients, you stir, you wait. Once the reaction is exhausted, then you remove your product and you throw away the residuals, okay? This is how you do every fermentation process. This is how you do yogurt, right? Do you need this for RNA, you don't. You don't need to use living bacteria. You don't need to use living organisms to do this. It's an enzymatic process, IVT. So what you can do this in flow. What happens when you do it in flow? Two major consequences. One, it's multi-scale, because the length of the flow path gives you the hold-up volume that gives you the scale. But it's the same identical process that happens because it's the same identical physicochemical conditions. In a huge tank, you have heterogeneity. In a flow path, it's all the same and can be simulated on a computer with incredible accuracy, with fidelity to reality. The second thing is that you can easily remove your product as soon as the polymerase, which is the enzyme that is reading the DNA template, as soon as it gets produced, which is great for quality, because most of the quality issues that we have with RNA production today come from the fact that the RNA, once produced, remains in that pot. And, for example, gets read again from the polymerase that double strands it and other things. And you have multiple quality issues. Now, today, the entire process to go from a computer-designed product to bulk drug product in the end is made of 19 steps. The first 11 are outside of GMP control, because that's for plasmid preparation, DNA preparation. And then you have the remaining steps within GMP. Okay? That's how you do it today. In the program, we have developed a number of different approaches. I'll show you two. The first one of which brings this down to 5 steps. 2 steps for DNA preparation without involving any living organism, no cells involved. And then 3 steps to get from DNA to the product. So the DNA part is developed by a company based in France called the DNA Script. What they developed is 2 units, desktop, like one is of this size and one is a little larger. So with two benches like this, you can host the whole thing. And in 36 hours, you go from computer-defined sequence to assembled and amplified DNA, ready for IVT, at a quality that is becoming comparable to that of colony picking. We are now at a 1 in 15,000 error rate. The company is targeting 1 in 20,000 for the next 5 months. And this is going in front of the US FDA for the first discussion on January the 7th. What do you do when you have DNA in your hands? Well, this is one thing that you can do. You can use the technology developed by another company called Centillion, based in Cambridge, UK. They have these 3 boxes that you can see here, 1, 2, and 3. 1 and 3 are actually the same box, but with a different cartridge, a different removable element here. Here you do IVT and pre-purification. Here you do RNA purification, and here you do encapsulation, formulation. Now, what is crazy is that you can use the exact same technology, multiple products. This has been tested on 15 different constructs now, different lengths, etc. I'll show you something. But what is crazy is that you can do exactly the same thing from a 10 milligram scale, like a sizable mouse experiment, up to 50 grams a day. 50 grams a day, if you think of a self-amplifying vaccine at a 1 microgram dose, that's 50 million vaccine doses in 1 day. Then good luck with filling that, but that's another problem, okay? And this has been now, this is going to UK MHRA on December the 6th with 1 product, and there's another one in development. Let me show you the second technology option. In this case, we have the DNA part that you saw before, without the amplification piece, only the printing, and then all the remaining steps all in one. And this is what Gary was referring to before. This is the technology developed by RiboPro. What you see here is a desktop unit, this one here, where you see a lot of electronics. This is the first prototype. Now we are at the third iteration. It's much slimmer. 20 of these units go into a vending machine like a Coke dispenser. It's the same form factor. It's 1.2 meters by 1.2 meters and 2 meters tall. Each of these units contains one of these cartridges here. This guy is called Mark. Very nice guy. You see this plastic layer here? Inside this plastic thing, you have DNA amplification, DNA purification, IVT, RNA purification, encapsulation, sterile filtration, which means that you go into that cartridge with a small scale of DNA and you get out of the other side bulk drug product ready for filling. Now, each of those cartridges can do from 10, now they're going down. They're going almost to 1 milligram, which is the most used sites for preclinical early studies. From 1 milligram to 1 kilogram a day. Because each unit can do 50 grams, 20 units give you a kilo. Or, if you don't want all the units to do the same thing, you can get 100 different products per day because you can do 5 cycles per day with one machine. Okay? And this is going to US FDA on January the 7th. How do we know that these things work? The only thing to know that manufacturing technology works is when you try to produce a product. There's no other way. So we worked on initially 20, then invested seriously on 9 products. We had great data here with the Staph aureus vaccine from Oakland. Then we selected 4 products that were ready for clinical development. And these are a self-amplifying rabies vaccine, an mRNA anti-COVID monoclonal antibody that I guess Phil will talk about because it's his product. And then we have another self-amplifying vaccine against COVID that is already in phase 3, but with a traditional manufacturing technology, and an mRNA anti-cancer product. The idea is to show that these technologies can manufacture very different products using exactly the same process. Same machine, same everything. You change nothing. Okay? And all of this with great quality in terms of integrity, minuscule double-stranded RNA, great encapsulation efficiencies, great control on the particle size and PDI, good potency outcomes. So there's 3 first-in-human studies. 1 is a phase 3 extension, and that's how we're planning between next year and the following to validate clinically the technology. And now the third piece. The third piece is, remember, the key event in computers was the rules, the new rules, how to democratize access to RNA manufacturing. And this is the last part of the presentation that I usually start with this question. Do you know how many atoms do we have in the observable universe? Who knows? No one? Yeah, it's an estimate, obviously. 10 to the? 80. Incredibly close. It's 10 to the 82. And given that it's a rough estimate, I would buy 10 to the 80. Can you think for a moment all the atoms in the universe? 10 with 80 zeros next to that. How large is that number? Now, let's ask ourselves, if I have a protein sequence, right? How many different DNA sequences encode for the same biologic, for the same protein? Identical amino acidic sequence, how many different nucleotidic sequences encode for the same? Now, if we take the spike antigen that we all received in our bodies, the number is 10 to the 632. So it's 500 orders of magnitude more than the number of atoms in the universe. That's the redundancy of the genetic code. Now, from a design perspective, this seems an unsolvable challenge. How the hell do I know, among the 10 to the 600 and something options that I have, which is the right one that I, as a product designer, have to choose to try and develop a new product? But if you think about it for a moment, this is a great opportunity. It's not just a challenge, because this means that with all these different options, you can optimize for multiple targets. So you can optimize for manufacturability of your sequence, and at the same time optimize for the biological functionality of your sequence. You want a product design that will manufacture very well on your line, so that you can keep your standardized line and you don't touch the manufacturing. Manufacturing is always the same. You never adjust it. You adjust the product. But at the same time, you can optimize the sequence also to launch well in cells, to be well translated, to do what you want once the RNA is delivered to the target that you have. I'm going to show you real quick two areas that we worked on this. One is the manufacturability part. We ran a 1 million RNA experiment in a wonderful collaboration among 7 teams of the 19 that you saw, particularly led by a team in Singapore at A-STAR and a team in Siena, Italy, Toscana Life Sciences Foundation. We synthesized a library of 1 million DNA sequences. We IVT'd, so we manufactured these million DNA sequences into RNAs with 5 different manufacturing processes. We did next generation sequencing using direct RNA sequencing and DNA sequencing with nanopore technology, and we developed a deep learning model that learned from the sequencing data what makes a good RNA product from a DNA sequence in terms of manufacturability, integrity, yield, the quality of what you get. And we have derived a predictor that predicts manufacturability with more than 80% accuracy, identifies the constructs that would be, how do I stop you? Yeah. There would be a problem for manufacturing with 90% accuracy, and without having ever been trained on a T7 terminator sequence, a canonical terminator, it can recognize nucleotide by nucleotide where a canonical terminator sequence is. It's amazing. It's borderline scary. Okay? So this is what we call now the VLSI rules of RNA, the universal manufacturing rules for RNA. The other space is how do you know that the RNA you are producing works? And we did some interesting work here with the team in Oakland as well. This is data from a team in Cambridge University and Kent University. What you see in pink is, this is a COVID vaccine delivered to mice. What you see in pink is the results you get using the Pfizer's optimization algorithm. We do not have access to the optimization algorithm, but we know what was the result of that optimization because we know the sequence they used, so we used that sequence. Or you see in blue the results that you get both in terms of neutralizing titers and functional CD8 percentages. You see what you get when you optimize differently with the codon optimizers that these guys developed based on tRNAome abundance in different tissues. All of this is being integrated in a portal that is launching at a not-for-profit institute called Pete Bioforge now in the US, and it's slowly offering runs with the technologies that you have seen and using these standardized rules for product optimization. I believe towards first, second quarter next year, most of the technologies that I've shown you should be available on that portal. Now, the final question before leaving you is, where do we go from here? Now, the program, this is the story of the R3 program, and that program is closing now. So we've been reflecting with, within Wellcome Leap and with other funders how to deploy this to the world. And I still don't know how that reflection will go, but what I can tell you is what we are discussing. And we are discussing the possibility to select a few locations in the world where we could deploy this technology to build biofoundries that would be like the TSMCs of RNA, biofoundries that only produce, that don't have their own products, that produce for others, without asking for any interaction with the IP of the product, because it's a pure CDMO model, with the difference that there's no adaptation to be done. The process is ready. If you follow the rules, you click a button, and you get your product. And it's the same process for a mouse experiment or for a clinical trial. There's no, okay, now I go into humans, let's make sure that I have a good GMP process. It's the same thing. Only the level of documentation is different because you don't want to produce all the paper that you produce for humans when you go in mice. But that's the only difference. And what is very important is that each of these locations would have contracts with the respective government so that when a pandemic happens, there's nothing to be invented. You have manufacturing capacity, it's there. We hope that someone will come up with a good anti-pandemic vaccine. By the moment that product is available, it's a click of a button, and you can get 10 billion vaccine doses in 1 week. So in a month, you could have enough vaccine for multiple doses for the entire planet. And manufacturing would be distributed in different geographical areas, so you wouldn't have the global access problem that we have today, because we know borders get shut down when you have pandemics. And this would mean, in short, saving millions of lives, never seeing again those military trucks that I showed you in the beginning, trillions of dollars that we have all paid as a financial impact of the pandemic, and enabling real global equity. Just to give you a sense, one of the cancer products that I was showing you that is in development now, the equivalent biologic has a price of approximately $10,000 per dose. The cost that we have, it's cost, it's not price. But the cost that we have in manufacturing, the same dose is $10. That means that the most expensive products that today are only available for high-income countries could become available for the entire planet. And with that, I'm done. Thank you.